one more time, good evening, everyone. And in our research group, we have a long standing interest in understanding how human insulin-producing beta cells are made. What are the molecular mechanisms that are controlling this process of their formation, but also their function? And we also have increasing efforts in understanding what goes wrong with these beta cells in cases of atypical diabetes. Okay. So actually, my interest in this kind of directions started with my postdoc. I was doing my PhD on mouse genetics and liver regeneration, and then when I graduated shortly after, the first human embryonic stem cells were isolated, and I thought about that this is such a great opportunity for whole regenerative medicine. And diabetes, especially type 1 diabetes, seems to be excellent target because simply one cell type is missing, the beta cells. So if we could understand the mechanism that will lead to the production of these cells in vitro, then in principle, we will have a great opportunity to provide new therapy options for the patients, but also more importantly, maybe from the researcher point of view, a great system to study what goes wrong with human beta cells during early stages of diabetes. So when we started, actually, this was quite an adventure, I have to say. And the efforts that initially were thought that would take like a couple years took collectively all the different research groups around the world. Around 20 years to learn how to make a good beta cells in vitro. And part of the reason is that we had to kind of agree what would be the best strategy. And let me only turn on the laser pointed. After some trials, we kind of collectively agreed that the best would be just to follow the key events of development in vitro conditions. So in order for the stem cells isolated from the blastocysts or the same from the induced pluripotent stem cells, in order to become pancreatic beta cells, it first has to become a definitive endoderm. Then it's specified into the gut tube pool of progenitors. Then they adopt a specific pancreatic fate. Their potential is then narrowed down just to only endocrine progenitors. And then the first immature beta cells are forming, and then there is a phase of maturation. And during this process, we not only have to agree what are the key steps that we have to achieve, we also kind of have to agree how to distinguish where we are along this road. So what will be the markers that allow us to navigate through this process? So you could imagine that each of the steps, it's multiple publications, multiple groups working, and so on, so on. But of course, this process it's also way more complicated than it was on this first cartoon. And we kind of quickly were able to work out the first stages, generating definitive endoderm and this early gut tube progenitors. But later on, what was happening is, first of all, that the efficiency of making these cells, these different populations of cells in vitro, was significantly dropping. So we could make definitive endoderm with efficiency 95 or even higher %. Here we systematically were like dropping with our percentages of success. And also you could see that multiple signals have to be either activated or repressed. These are shown in the brackets. And with the development of all these different single cell omics techniques, we quickly understood that what we have here, it's a really heterogeneous mixture of different cell populations. And as a result, what is happening, that in a dish, we have a mixture of cells. not only we have the beta cells, we also have beta and alpha cells. We also have cells that do not exist in pancreas, so-called enterochromaffin cells, which more resemble the population of cells existing during development of intestine. We also have a bit of acinar cells and ducture. So when I started my own research group, the first question we ask is how these decisions are made regarding the islet cells, because we knew from the years of development of biology that this population of the 5 different islet endocrine cells, they are all coming from a common progenitor. This progenitor, we call them endocrine progenitors, and they are marked by the expression of master regulator transcription factor neurogenin 3. So the question was, we have to make neurogenin 3 positive cells, but then when and how these cells make the decision whether to become an alpha or beta cells. And okay. And what we decided to do, and this was just truly the beginning of the single-cell RNA technologies, we decided to sequence the murine pancreas at 2 developmental time points where most of these neurogenin 3 positive cells are formed, and hence most of the alpha, beta, and so on cells are formed. So you can see here the overview of the whole pancreas. So you can clearly see different populations, including we got like 8 hepatocytes and different mesenchymas. Of course, these hepatocytes are kind of contamination of our prep. But I like it because they also kind of a sign of how sensitive this whole system is. You have mesenchymes, you have some endothelial, and you also have these neurogenin 3 positive endocrine progenitors. So when we had a closer look at these specific populations, we were able to distinguish 4 molecularly distinct subtypes of endocrine progenitors. And we were very happy about this result because we initially assumed that, of course, one of them will be the future beta cells, one of them future alpha cells, and so on, so on. But to our surprise, it turned out not to be true. These 4 different subtypes did not reflect future hormone-producing cells, but rather it reflects different kind of maturation status of these endocrine progenitors. So looking at the pseudo-time analysis, we're able to identify the youngest, the earliest population, and then a bit of outsider, middle population, then EP3 and EP4. And if we look at different genes associated with these subtypes, we're able to say that this represents the early progenitors. Some of them were also like the bipotent, have markers of the early, even earlier progenitors, bipotent progenitors. Then these cells were undergoing the delamination. They were leaving this epithelial cord of pancreatic progenitors and kind of to form in the future islets in the surrounding mesenchymal and endothelial cells. Then we had something which maybe we'll call a true core endocrine progenitors, and then these late endocrine progenitors which already started to upregulate markers characteristic for alpha and beta cells. What was great is not only that we could kind of link each of these to different stage of the maturation process, but we were also able to identify a cohort of genes that had a very characteristic pattern at each stage. So for example, in this EP2, we are able to identify a group of genes that were highly upregulated just in this cluster, but not in other subpopulations. And these were genes associated with epithelial mesenchymal transition or the mechanosignaling like YAP. And although this work was done in mice, we were later able to confirm that the same subpopulations and the same kind of gene expression dynamics we could significantly recapitulate in the human developing pancreas. Okay, so this was our first conclusion, that we have different subgroups of endocrine progenitors, but they do not reflect future alpha, beta, and so on cells, rather the stage of their maturation. Then the question still was like, then when and how alpha and beta cells decide whether they will be these cells, right? So where we put together the neurogenin 3 positive cells from the early time points and the later time points, we noticed that majority of them do not cluster together. They form two different populations. So we kind of analyzed them deeper using a cohort of methods. And it would turn out that the EPs that were born at the early time points at day 14 of murine development, they were more prone to become alpha cells. They were more likely to upregulate alpha cell specific markers, while the endocrine progenitors born 2 days later, they were statistically more likely to become beta cells. And we also confirmed this with another unbiased method looking for the chromatin accessibility. And again, the mode of enrichment suggested that the earliest EPs are more associated with alpha-specific transcription factors, while the late EPs more with the beta-specific transcription factors. So what we concluded from this work is the following, that the EPs born at those different time points, they all express neurogenin 3, but the early EPs, and maybe that's worth to mention here, that these EP cells, these neurogenin 3 positive cells, are kind of short-lived. They don't stay for long. Approximately after 24 hours, they become something, they adapt to the next fate. So these EPs that were formed earlier, they are more likely to become alpha cells, while the EPs born later, they are more likely to become beta cells. So we were trying to understand like why, what's really changing except this time, during this time window, that changed so much their differentiation propensity. And one thing that kind of pops immediately to our attention was that during this time, there's a massive morphogenesis reorganization of the whole formic pancreas. So what you can see here in this purplish, it's this pancreatic epithelium, which of course it's surrounded by formic mesenchyme, endothelial, and incoming nerve cells. And here you can see in these green dots, those endocrine progenitors that are leaving this epithelium to form the future islets. And the epithelium, it's far less complex, but 2 days later, it's a huge branching reorganization events. Also, the tip cells are forming, which will form the future acinar cells and so on. So we kind of focus on what's going on not only inside of these endocrine progenitors, but also in their surroundings. Okay, and we particularly focus on one subpopulation, which we found very interesting, and these were those EP2. Why? For one, they were really truly molecularly quite distinct from the rest of endocrine progenitors. For two, they have a very specific group of genes that are differentially expressed in this subtype. And these were like all the genes associated with cell motility, migration, mechanosignaling, and so on. So we thought that because these endocrine progenitors, they have to leave this pancreatic epithelium, and because we truly didn't know what is the mechanism and what are the kind of regulators of this process, that this subpopulation might reflect, this might reflect the cells actively kind of undergoing this rearrangement. So this was also the time when I moved back to Poznań, and I started with new group of young people. So basically, what I asked them is I gave them all this data, and I said, for each of them to select a gene that is enriched in this endocrine progenitors subtype 2. And then let's see what happens where we manipulate its expression level. So we selected quite random genes in the sense that there were transcription factors, there were like cytoplasmic enzymes, there were also genes clearly associated with the cytoskeleton, cell cortex, but also like ECM associated proteins. And we just one by one study what's their function during this particular period of beta cell formation. And although, you might think that this whole in vitro stem cell differentiation business, it's definitely not a perfect system, right? Because we miss a lot of things that are going on in vivo. But also, it allows us to study multiple aspects, including some aspects of the morphogenesis of this cellular rearrangements. Because as you can see, we differentiated our stem cells as a 3D format, as those kind of clusters. We call them usually spheroids. And then you could see that during this whole process, if you just take the Y-type cells, there's a lot of going on. So at the stage around the primitive gut tube, you can see this clearly budding events. And actually, we know what kind of markers will be highly expressed in these buds compared to the core of the spheroids. You can see that 2 days later, the cells which kind of were budding, they are kind of incorporated again inside of the spheroids and so on. So even this somehow not perfect system allows us to delineate at least some of the parts of the cellular movements and rearrangements. So the first gene that I'm just want to briefly show you today was the story about the transcription factor ETV1. And when we knock it out, and this was also, I was very hesitant to start this because it's one of the first transcription factor that ever was knocked out in mice. It was a work by Sylvia Arber in Basel, and it has also a phenotype associated with the neuronal development. And I thought, what new we can still learn about this gene? But nevertheless, we tried to do it, and it turned out to be worth our effort. So what you can see here, that even looking at this morphology of the spheroids, they behave quite differently. The initial stages were the same, but later on, you could see that this budding process, this making those rosettes, did not work so well at the day 6. And then later on, where the spheroids should have more or less circular form, then the cell seems to be very irregular with those different outings. And if you look later on, you can see that they look even worse and worse as the time of the differentiation progresses. We could also quantify this. So, at this PP stage, you could see that the organoids, they are bigger. The knockout organoids are bigger. They are also less round, and their circularity is also changed compared to the Y-type controls. So this will suggest that this morphogenic events are not happening at the same way as it is in the Y-type cells. So when we follow it up with the marker of different stages of pancreatic differentiation, you could see that the early stages making definitive endoderm, everything is perfect. But then later on, we have the initial pancreatic specification. You can form these early pancreatic progenitors, but the longer you go during differentiation, then these pancreatic progenitors do not mature properly, do not upregulate the expression of another key marker, the transcription factor NKX6.1, and then no chromogranin endocrine cells form, or at least with very low efficiency, and as a result, we don't have really much of the C-peptide. So then we decided to do a single cell and look more closely at what's going on with this knockout versus Y-type pancreatic progenitors, just at the stage where we saw this clear phenotype. And then when we look, different clusters, more or less the similar clusters were formed in both wild type and knockout cells, but the frequency was different. So we had an overrepresentation of these early progenitors, consequently less of more mature progenitors. We have less of endocrine progenitors and endocrine cells. And we had some more of mesenchyme and liver-like cells form, which we actually did not follow it up, but it also might be interesting in the light of another project. Yeah, and then if you look closely at this different population of pancreatic progenitors, you can see how dynamic period it is. So these early pancreatic progenitors, they have a tons of different signaling going on, and a lot of them will be associated with adhesion, cell-cell communication, migration, motility. So that's really a busy time in the life of pancreatic progenitors. So when we compare the route of wild type and knockout cells towards the endocrine progenitors, you can see here that the wild type cells were systematically progressing towards the endocrine cells as it's supposed to be, while in the knockout we have initial process going well, and then they kind of stumble, right? And then only a smaller group of them is moving forward. And you can also see it here, if you look at this pattern, that's the wild type cells. We have here the proliferating branch coming off, and then they go towards the endocrine cells as we actually lead them through our differentiation process, right? Giving them different morphogens, different signaling molecules to kind of push them towards these endocrine cells. While the knockout cells, some of them reach this endpoint, but they are kind of moving into circles. This is definitely not a straight way. And then the ATAC-C kind of confirm our differentiation defect that there was no enrichment for the PDX motif, which would be necessary for these pancreatic progenitors to mature further. And then the loci for many genes show different accessibility. Many of them were the key transcription factor for the pancreas and beta cell development. For example, this neurogenin 3 that I mentioned to you. But also many of them were associated with other processes. So when we look particularly like what's really wrong with them and with the knockout cells, it's basically whether we look at the early or at the late pancreatic progenitor population, the same kind of theme was repeating. Focal adhesion, cytoskeleton, axon guidance, PI3 kinase, MAP kinase pathway, everything was kind of repeatedly being upregulated in different subtypes of the knockout cells. And we could also confirm this on the protein level, and I'm showing you several examples. So for example, a vinculin associated with the either cell or ECM adhesion markers were highly upregulated in the knockout cells. Here you have a higher resolution microscopy showing it more clearly. The same was with the collagen 4 and the integrin. And this changes in their production of protein associated with communication of cells, either with their neighbors or ECM, kind of change their ability for this morphogenetic rearrangement. And hence they could not properly rearrange themselves and they could not properly differentiate. Okay, so this is basically showing similar thing, just the quantification. And as you can see also on this sagittal section through these spheroids, their cytoskeleton and all this kind of adhesion-related molecules are completely messed up in the knockout. So what I'm showing here is the F-actin in green, and then navigator 3, which is associated with the microtubules. And you can see that already at the day 12, the pattern looks quite different in the knockout. We have a higher nodes of concentration for F-actin. The navigator is also not properly localized in the cells, but at later stages, it becomes even more pronounced. So look here in the stage where the endocrine progenitors are formed, where we have again these buds forming that have to then kind of be incorporated again back to the spheroids. There is a clear ring of F-actin forming in the knockout cells, and this kind of inhibits this possibility of this rearrangement. And at day 24, where the early beta cells are, then this rearrangements kind of calm down, and we have quite uniform distribution of F-actin and navigator 3, but not in the mutant. In the mutant, we have a situation that kind of resembles like a grapes, where we have those little rings around every cell, almost like they are kind of separate entities. And it's not only about the ECM, I'm just showing you one of the examples, but multiple signaling pathways were also misregulated in the knockout cells. We have here SLIT, which is part of the SLIT-ROBO axon guidance signaling, but also it's just basically regulates the motility not only of axons, but also of other cells. And you can see that in the knockout, we have a higher abrogation of SLIT3 compared to the wild type. Okay, so this changes. So this was interesting because this was one of the rare examples of what is the transcriptional regulation of cell way to communicate with their surroundings, right? So we could show that by manipulating the expression of the transcription factors, we can change the production of cell and cell ECM related proteins, and then change the whole mechanosignaling, and as a result, this will affect the cell fate acquisition. So we, of course, know that the ECM, it's important, right? We know that by regulating the stiffness of composition, we can change the mechanosignaling, but we show it also a bit from the other perspective. We show what kind of transcription factor inside the cell regulates its way to communicate with its neighbors. Okay. So let me briefly move to another player that turned out to be quite interesting, and that's an glutathione peroxidase, an enzyme that is involved in dealing with the reactive oxygen species, and it's part of the system of a response to the oxidative stress. So, when we started the project, I would never assume that this has anything to do with the differentiation of endocrine cells in the pancreas. But what was quite interesting is that if you look at the expression level of ROS during the development, you can clearly see that it's increasing. And of course, we know that oxidative stress, it's critical for adult beta cell function. And we know that the excess of the oxidative stress can lead to the dysfunction of beta cells or even their death. And we also know from other studies, multiple studies, that oxidative stress, it's not only negative, right? It can also act as a signaling molecule. So we try to understand, is there any meaning behind this increasing level of reactive oxygen species during beta cell development? And then, of course, there's multiple enzymes that are involved in this response. So we try to look which enzymes are well expressed. And one of the enzymes that turned out to be interesting, it's GPX2. It was highly upregulated compared to the other family members or other family members in the pancreatic progenitors. Along with the SOT3 was also highly upregulated in the same pancreatic progenitors population. However, because the GPX works downstream of SOT, And because also we knew from our previous studies that GPX2 is so highly upregulated in this EP2, we focus on those genes. You can also see that it's nicely expressed in the human fetal pancreas around the time where these endocrine progenitors are forming. So that's why we decided to knock it down and do overexpression. We are always doing this both ways regulation. And we look what is happening with the beta cell in vitro formation. Early stages, everything works perfectly. But already at the primitive foregut stage, we could see something quite surprising, that it seems like the cells are different, the knockout cells are differentiating better. So you could see they kind of produced more PDX1, more another pancreatic marker SOX9. But what is interesting is that, of course, SOX9 is not only expressed in pancreas, it's also expressed in the liver. And then when we look in other markers associated with this primitive foregut differentiation, like another liver marker, intestine marker, we could see that also they were increasing. Then the hex marker, that is inhibitor of pancreatic differentiation, was a decrease in the knockout cells. So as a result, what we kind of concluded is the following thing, that in the absence of GPX2, which, by the way, led to the increased oxidative stress. I'm not showing you this data, so you have to take it for my word. But what is really happening is that the late endoderm accelerates in development, but despite the fact that we are specifically pushing them towards pancreas and beta cells, they are upregulating also markers of liver and intestine. So this was quite unexpected. So we wanted, with the immunofluorescence, we can only look at the handful of different markers of different proteins. Of course, we cannot test tens of them. So then we wanted to look at this in more a bit unbiased way using the RNA-seq, either population or the single cell RNA-seq. And we did this on different stages. I mean, here the differentiation is quite efficient, so we have quite homogeneous population. At the later stages, it makes more sense to actually do the single cell, because as you can see, we have a different kinds of progenitors present at the same time in a dish. But nevertheless, each of this experiment pointed out to the same thing. You could see that in the knockout cells, we have upregulated genes associated with intestine, with liver, the BMP signaling, and so on. You could see also that there is suppression of pancreatic differentiation program at later stages, that there is suppression of hypoxia, reactive oxygen species, and then we have also the changes in fatty acids metabolism pathway. And when we look a bit like what kind of cells do we have in the dish? So we look at day 21 of the differentiation. It's the day where we have already some beta cells forming, right? So, and in this orange shade, you could see the white type cells, and in the blue, the knockout cells. What you can see here, if you merge these two data sets, that there is some overlap clearly, but there's also quite big distinction between wild type and knockout cells. So these overlapping cells, they were representing endocrine cells and alpha and beta, delta, alpha, and multihormonal cells. And then here we had a group of cells that were specific only for the knockout. And when we look at the markers that what these cells are, we name them that these represent and multilineage progenitors 1 and 2, and again, the multilineage cells. And when you look a bit closely, you could see that there is a cohort of liver-specific genes that are upregulated in this population, this multilineage population, and also even in this beta cells or endocrine cells. And these upregulation, it's stronger in the knockout compared to the wild type. And again, if you look at the pseudotime and try to track each step of this differentiation path, you could see that we have the pancreatic progenitors moving towards endocrine progenitors. I mean, here, this dark green, these are proliferating pancreatic progenitors. So, of course, they're a bit aside, but we have pancreatic progenitors moving towards endocrine progenitors and then endocrine cells. And here we have the pancreatic progenitors, some of them moving nicely towards beta cells, and some of them going sideways and forming this weird liver-like, intestine-like cells. And just please keep in mind that this is everything under conditions where we are promoting beta cell formation and inhibiting, because we are also using inhibitors of intestinal cell fate and liver cell fate and so on. So we looked a bit closely at this, whether this is really something to do with the oxidative stress. And okay, so if you take Y-type cells and you expose them to H2O2, you could see that they do indeed produce more PDX. They seems to be kind of more prone to become pancreatic progenitors. And what we could also use as a marker of this treatment was that the H2O2 is also changing the structure of the ECM, and here is shown on the collagen fibers that they become disintegrated. And then when you take the knockout cells, you can see the knockout cells, they are again producing more PDX. That's what we saw. And it's kind of similar what it does, the treatment of H2O2 with the Y-type cells. And when you treat the knockout cells with the antioxidant, then you could at least partially reverse this this precocious pancreatic differentiation. And surprisingly, you can also reverse this ECM collagen fiber disintegration, right? By simply treating the cells with the selenium. So it seems like that the GPX2 deletion causes increased oxidative stress, and then this oxidative stress is making cells to differentiate faster and in different lineages despite the pancreas promoting condition. And yeah, and this oxidative stress seems to be kind of important player in this whole process. Yes, so we also kind of look through different systems using overexpression and so on, and we also did the ATAC-seq, and you can see the liver markers being more accessible in the knockout cells, FGG, one of the CYP enzymes. You can also see on the protein levels the FGB being highly expressed in the knockout cells. And so on, but what we also did here, maybe what I should mention at the beginning, is that here we moved away from this directed differentiation towards pancreas, but we asked we said this: if our knockout cells, when we tell them to become beta cells, they are, at least some of them, becoming liver-like cells, what will happen if we removed any pancreatic promoting signals from our medium? So we differentiate them towards definitive endoderm, and then we just add base media without any specific signaling molecules. So what you can see is that the knockout cells, they made a lot of liver-like cells. We did not see any PDX expression, and you may say, oh, but previously the knockout cells were doing more PDX. Yes, but to make PDX, you need actually activate retinoic acid pathway. And this is one of the signals we are adding to our differentiation media. And of course, this signal was missing in these conditions. So we did not see any PDX, but that's kind of to be expected. We again saw more CDX2 intestine marker in the knockout cells. And you can see here also at the lower magnification how much more the knockout cells were making. And then where we compare, when we look at the panel of liver specific genes in the knockout versus wild type cells, you can see a lot of the liver specific genes being upregulated in the knockout cells. If we compare the wild type cells treated with H2O2 versus wild type, again, many of these genes were also upregulated after the treatment with H2O2. So it seems like that this H2O2 is inducing the hepatic sulfide alone without the GPX2 deletion. And where we compare these two conditions, you can see that, of course, many genes are still kind of being dysregulated. And you could say this might be due to the treatment, the length, the concentration, but also we might need to add another signal to fully recapitulate the GPX2 deletion. Okay, so we knew that GPX2 via oxidative stress is causing cells to more likely become liver-like cells in vitro. So why? How is this working? And then one of the things that really got our attention was the changes in the ECM. Because in the knockout cells, we have a lot of genes associated with the ECM dysregulated. And we could not only see this on the mRNA level, also on the protein level. So LAMA1, LAMA5, collagen fibers dysregulated in the knockout cells. And also what we noticed was this dysregulation in the TGF-beta regulon, which was clearly more active not only in the knockout cells, but especially in this multi-lineage clusters. So in our analysis, the TGF-beta regulon, it contains the effectors, the genes associated with the BMP and TGF-beta pathway, both pathways that are very much known for their differentiation potential, right? That they can affect the differentiation of multiple cells. So we treated our wild type and knockout cells with either activator of the BMP or TGF-beta or inhibitors of one or both of the pathways. I'm just showing you kind of summary of this experiment, and it was the following. And if we look at the expression of one of the liver markers, what was happening is that in the wild type cells, when we treated them with the BMP activation, then we have the upregulation of this liver marker expression, similar to the level that we have in the knockout cells. And when we treated knockout cells with the inhibitors of BMP and the inhibitors of TGF-beta signaling, that we could see the suppression of the HNF4 alpha, the liver marker expression. So together we concluded that the deletion of GPX2 is increasing the ROS levels, the cytoplasmic ROS levels, which is causing the changes in the ECM. And ECM, it's not only collagens and laminins, right? But multiple signaling molecules, the ligands and so on are deposited in the ECM. And these changes are kind of releasing and activating them. So we can see the activation of BMP pathway at the higher level in the knockout cells, which we also confirm by looking at different SMARTs and so on. And this will push our cells, even in the pancreatic conditions, to adopt liver-like fate. So that was kind of interesting because just looking at the genes that were upregulated in the subpopulation of endocrine progenitors, we were able to identify some of the genes that can modulate cell fate through the cell communication system, either through the neighboring cells or with ECM. And these could be genes working in different level of cellular organization, like transcription factor or the cytosolic enzymes. But either way, messing up with this mechanosignaling will change the cell fate. Okay, so let me now switch the gears to something a bit different, just to show you a bit of different taste of what we are doing, is that going back to this endocrine progenitors and their formation during pancreas development, you have here here what I'm showing is you a cartoon of kind of a snapshot of what's really going on when these endocrine cells are forming. So we have in this bluish and red and yellowish the pancreatic epithelium with different kinds of progenitors, future acinar cells, classical pancreatic progenitors, and then some of them will become a future duct cells. And among these pancreatic progenitors, some of the cells will upregulate neurogenin 3 and become endocrine cells, which will delaminate this pancreatic epithelium and then will form an islet. And of course, that's not only about this pancreatic epithelial cells, we have also the endothelial cells coming in, and we have also the mesenchymal cells forming and neural crest cells, and so on and so on. So because of this morphogenetic changes happening around this time where these islets are starting to form, we also wanted to have a bit closer look, not only on these cells, but also on their surrounding. So basically, we reanalyzed the same data sets from the single cell of developing mouse pancreas. And we were able again to identify different subpopulations of mesenchymal cells, of course, different markers and so on. So we try to understand what is the kind of meaning behind this diversity among mesenchymal cells. So we kind of designed this whole evaluation system where we do the analysis of the interactome, just trying to see which subtype of mesenchyme can crosstalk with each others, with what kind of other cells that are present in developing pancreas. We also did the pseudo-time, and then we did the validation using lineage tracing and so on. But what I mostly want to show you today is this what we called a spatial mapping approach. Okay, so looking at this interactome analysis, we could, for example, see that these mesenchymes, they seems to have certain propensities. So mesenchymal 3 and 4, or mostly actually 4, they had a lot to do with the innervation. The geo-enrichment will show a lot of terms associated with axon guidance, innervation, and so on, so on. But also we have the EMT, the cell migration, and so on. So when we did the light sheet microscopy of developing pancreas in different stages, and we stained these different mesenchymes and main components of pancreas. So the ACE2, it's marker also of this MES 3 and 4, mostly of MES 4, and hence also how we started to work on ACE2 and SARS and COVID with Krzysiu Perć some time ago. But here, coming back to mesenchyme, what we could see is that it's not easy to kind of analyze those images, but we did it like we really spent months, if not years, doing this analysis. So you can see here the early pancreatic bud. There's not too many of ICE cells, ICE2 positive cells. There are some here if you would zoom in. But later on, you can also see some innervation forming, right? You can see the nice projection marked by the TH. And then you can also see some ACE2 positive cells. And if you look again closely, here's another time shot, you could see that a lot of these ACE2 cells will localize along this forming innervation. And we, of course, did the analysis on more like zoom in pictures. And we also kind of developed a system to quantify this colocalization. And what it turned out is the following, that the ACE2 mesenchyme marked by ACE2 mesenchyme, which represents mesenchyme 4 subpopulation, these cells preferentially cluster nearby the forming beta cells, insulin positive beta cells, or nearby the innervation. They hardly ever were somewhere else in the pancreas. And just to show you kind of maybe one slide that this the system has a bit of sense, makes a bit of sense, is I wanted to show you one slide on another mesenchyme subpopulation, which is as we call the mesothelium O, which is marked by the expression of WT1. And if you look at even on this UMAP plot, you can see that this is kind of outlier, right? It does not cluster with the rest of the mesenchyme. But if you look at the pseudotime, it seems to be the earliest population of mesenchyme, at least in our analysis. And also others people show this way, that this is kind of like a progenitor pool for these other subtypes of mesenchyma that are forming. So when we apply our system for the spatial mapping and quantification, what you can see here is that here we have an insulin positive cells and cadherin positive cells, E-cadherin, so basically marking pancreatic epithelium. And you can see that they nicely co-localize, which makes sense because this endocrine, this first beta cells are forming next to this pancreatic epithelium. So that's nice. And you can see that this WT1 cells are more far away from these two subpopulations. And if you would look at the stainings, you could see that these WT positive cells, they usually are outside of the pancreas. They are forming a thin layer outside of this pancreas. So just what I wanted to show through this is that this system are able really meaningfully to quantify or to assess the colocalization of different subtypes. So what was interesting is that actually, if you would isolate the ACE2 positive population from the mice and mix it up with the chromogranin positive cells, they will also, even in vitro condition, they will kind of find each other and cluster next to each other preferentially. And the same kind of colocalization we could observe also in human developing pancreas, so that this mesenchyme 4 clusters together nearby insulin and TH positive cells, as opposed to this WT1 layer, what you can maybe see nicer on this image that outlines the pancreas, right? It forms the layers outside of the pancreas. Okay, so we look at the interactome, right? What is that MES4 are secreting that receptors for these signals are present or either pancreatic progenitors or endocrine progenitors and kind of make the list. And this was interesting because one of the candidates, one of these pairs, ligand receptor pairs that we identify was the Wnt5a. And why this was interesting? Because this nicely aligned with another project that was ongoing in the lab. And in this project, we again try to understand what is the contribution of microenvironment in pancreatic development. Oh, I have to kind of finish up. Sorry, I just was talking too much. So let me quickly finish it up. But we did it with completely different approach. We generated mesenchymal endothelial cells from different stages of pancreas development, of human pancreatic development, and we co-cultured them with the pancreatic progenitors generated from the stem cells in vitro. And we were able to identify two specific lines generated from two specific time points, which kind of preferentially push these pancreatic progenitors towards beta cells, that more beta cells were forming in their presence. And then these beta cells also secreted significant amounts of C-peptide, and this was almost the same as human cadaveric islets do secrete. So then trying to understand what is this mesenchymal from week 17, head of pancreas, and mesenchymal from week 20 of pancreas producing, we kind of came up again to different, ligand and receptors pair, and we selected those and tested them one by one, just using recombinant protein. And one thing that kind of popped out was Wnt5a. So this was such a nice kind of crosstalk between different projects. And when we deleted the Wnt5a and this mesenchymal endothelial cells, it turned out that almost no beta cells are forming when we co-culture endocrine progenitors with these mesenchymal cells that do not have Wnt5a. So the Wnt5a seems to be important for this process. And we later established that how it works, that we know that initially the Wnt5a, after short time treatment, it will activate the Jun pathway, but after several days of treatment, it will inhibit the BMP signaling. And we established that this coordinate action of this two signaling pathway is important for making beta cells. Okay. So maybe just using a couple more minutes, I will quickly go through one more thing that I wanted to share with you. It's about, one thing that keeps bothering us for years is the efficiency. Because if we think about making a lot of beta cells in vitro, this differentiation process it's a bit tricky. It takes quite a long time. You get fully functional cells after 6-7 weeks of in vitro differentiation, and you have roughly 30, maximum 50% of beta cells in a dish that be forming, right? So 50% of the total cells in the dish are becoming beta cells. So you really need quite a lot of dishes, quite a lot of differentiation, which unfortunately are expensive to make a significant amount of beta cells. So looking at this simplified scheme, we know that we can nicely expand pancreatic pluripotent stem cells. Also during my postdoc, I showed that we can nicely expand endoderm. Like, why we cannot expand beta cells? Well, one of the problem is that the adult mature beta cells are famous for being very stubborn in sense of proliferation. And as you can see here from another studies, in the adults, way less than 1% of human beta cells are proliferating. But there is a window shortly after birth where there is a burst of proliferation, although there's only 2% of beta cells are forming. So this would suggest that there is something different about this early form beta cells versus adult, and maybe this difference will help us to identify signals that will allow us expansion of beta cells. So looking at this transcriptomics of these cells and that, we identify one of the ECM factor, which also was present in our EP2, which was SOX2. You can see that SOX2 is present in the human developing pancreas. It's also present in the in vitro derived cells. And where we knock down this SOX2, we can see that, and this we did in the cell line, the endo-CBH human fetal beta cell lines. We can see that we have an increase in proliferation. And where we overexpress, we have the opposite effect compared to the wild type. And when you look which cells are expressing the proliferation markers, you can see that the cells that have less SPOC, lower level of SPOC, they are expressing more proliferation markers versus cells which have higher amounts of SPOC, they do not proliferate. Okay, so it seems like the SPOC2 is the negative regulator of proliferation of early beta cells. And when we look at the molecular events associated with the downregulation of SPOC2, we identify one which was quite interesting, which is MMP2. And through these different experiments, we establish that by treating the cells with MMP2, because MMP2 was upregulated in the SPOC2 knockdown, we are able to induce similar effect in cells of beta cell replication. Okay, so you can see here that if we passage our stem cell derived human beta cells in the presence of MMP2, then we have way more cells, and this effect can be repeated over several passages. And when we look at the glucagon positive cells, we see no effect. So it seems to be specific for the beta cells. Maybe I was, okay, maybe what was also interesting is that despite the notion that beta cells either can proliferate or function, but never do both, we were able to establish that this MMP treatment of early beta cells, this freshly formed, also increased their insulin secretion. So you can see here, these are untreated cells, untreated with MMP2 cells, which were exposed to low glucose, and there's very little insulin secretion. But when we expose them to high glucose, there is a clear spike in insulin secretion, as it should be. And if we treat, but if we let the cells to keep them in culture for multiple passages without treatment with MMP2, this response is decreasing. But if we do the same in the presence of MMP2, then we can even get more insulin secretion stimulated by glucose. And we did this in vitro, but we also confirmed the same effect in vivo when we transplanted those human stem cell derived beta cells that were passaged in presence of MMP2 or without MMP2 into the immunocompromised mice. And what you can see here, which is very nice, is that as a control group, we transplanted the human cadaveric islets, and the MMP2 treated cells, they show similar response to human islets. Okay, maybe I'll skip this. And what I also want to highlight it, that this trick, the SPOC2 MMP2, does not work for the adult human islet. And it does not work for other cells. Okay, so let me maybe, I'm sorry that I talk so much. So let me maybe finished here. I wanted to briefly tell you about some cases of atypical diabetes, but perhaps I can leave this for the next time. Let me only tell you this, that we are very much interested in understanding the molecular mechanisms associated with atypical diabetes. And these, of course, it's done in collaboration. Actually, this is still done in collaboration with clinicians from the United States, which they formed the consortium that in multiple clinical sites identify the patients that have higher rates of ketoacidosis, and then they subcluster these patients into 4 groups, whether they have or don't have beta cells and presence of autoantibodies. And we are mostly interested into this subpopulation of patients which have no autoantibodies and also no beta cells, which are enriched in monogenic forms of diabetes, but also some of them are quite interesting cases. And we have so far published, case study for one patient, but let me maybe finish up with something more future. So we are now trying to establish, because, through several years of this Radiant, because this consortium is called Radiant work, we're able to identify 1,200 of patients with atypical diabetes. And for these patients, we have the metabolomics data, transcriptomics on their plasma cells. We have the whole genome sequencing and so on, so on. And our part of this consortium is make iPSCs and do the modeling of the beta cell development and function in vitro or using our in vivo model. And of course, we cannot do this in such a big number of patients the same way as we have been doing so far. So taking one patient and carefully studying the beta cell formation and molecular changes in different stages and so on, because simply we don't have such a capabilities. So we kind of came up with this idea. We have done some preliminary experiments, and now we are trying to secure some additional funding to be able to do it more precisely. Because another problem is that, of course, in each patient, our colleagues from Broad Institute were able to identify, several SNPs, and according to their algorithm, usually like 5 to 6 SNPs per patient seems to be promising in the sense that they might have to do something with beta cell development and or function. So, we are not able to study each SNP by one. So we're thinking about approaching this from a bit different way, where we take the Y-type iPSCs, we differentiate them towards pancreatic cells and beta cells. And then we will do the whole genome CRISPR-Cas screen, and then we inactivate number of genes, as many as possible. And then at those 2 different stages, we will make transcriptomics. So we will get the signature that deletion of this gene is associated with these changes in transcriptome and so on, and so on. And in parallel, we initially declared to do 20 patients that we carefully selected with based on the genomic analysis. And we will also differentiate, make the iPSCs, which we have already done, differentiate them towards PPs and beta cells, which we also have already done, and do the transcriptomic analysis, and then cross-compare, right? These 2 signatures in order to identify some similarities. So the idea is that we are hoping to see that, for example, the transcriptome of patient 1, it's similar to the transcriptome of knockout of gene number 30. And if this gene is one of the SNPs that we identify in this patient, this kind of allows us to narrow down where we should focus. So this will be our approach. And maybe what I also wanted to kind of close up with, just to show you that the in vitro derived beta cells are actually quite good. So what you can see here, the high resolution microscopy, you can see vimentin just to kind of as a background, but what you can also see here, nice insulin granules. We can also see them nicely in the distributed or accumulated near the membrane. We can also confirm this with electron microscopy. We can see the calcium influxes going on once we stimulate our spheroids with beta cells with glucose. And we can see quite nice secretion of either human insulin or C-peptide in response to different concentration of glucose. So you can see also this, what I was telling you before, that the time is of essence. So the early beta cells, they show smaller response. The later beta cells, they actually show quite good response, that it's almost the same. It's undistinguishable from the human islets. So I think we are in the good position to do really systematic studies of patient-specific cells, and we want to mix it up with this model where we take the immunocompromised mice, we transplant them the human stem cell-derived beta cells under the kidney capsule. It's not really major surgery. And then we kill off rodent beta cells with simply a drug treatment. And then we have the situation, if our beta cells are not working very well, if these are not good beta cells, then we will see hyperglycemia. But if we provide a good beta cells derived, then we should see the normalization and so on. And we also want to use the system to study also the patient cells as well. And also what is nice, because then we can remove the kidney with the transplanted cells, and this should lead that again the blood glucose level will increase. Okay, so let me just close up with some shout outs to, these are the people who are currently working with us, and those shown in blue, they were critical for the stories that I share with you today. So Wojtek Szlachtic, who is very talented postdoc, I think he's on a on the path becoming independent, and Asha work on GPX2, Natalia and Magda on ATV1, and Kasia on SPOC2. And the picture, I'm sorry, and our collaborators. And thank you very much for your attention.